Okay, so let's do maybe a detailed version of what's going on when a primary response happens, so that we understand how the B memory, T memory, antibodies, T killer cells, T helper cells, how do they all fit into this picture of the immune response. So if we have a few cells here, and we say that some of them have been infected, okay, and then we also have some pathogen which is outside of the cells. This could be a virus, this could be a bacteria, it could be anything. Okay, now what is a sequence of events that leads to the activation of the B cells and the T cells? So first thing is you have a pathogen and it is phagocytosed, yeah, by a macrophage. So first step, macrophage or other phagocyte, macrophage or phagocyte engulfs pathogen, could mention phagocytosis, and what they do once this whole process is done, they engulf the pathogen, the lysosomes fuse, they break down the pathogen, and they then present fragments of the broken down pathogen on their surface. Okay, so step one was the engulfing. Step two is antigen presentation. Okay, so the macrophage or neutrophil has broken down the pathogen within the cell, and whatever the products of the digestion was, the fragments of that pathogen are presented on the surface of that cell. Okay, this is called antigen presentation, and at this stage, this macrophage is called an antigen presenting cell. Okay, now we then have a range of T helper cells. We have loads of T helper cells, all with slightly different CD4 receptors or T cell receptors. Okay, and this is what they use to identify their infected cells. Okay, so I'm only showing 4 here, but we have millions of different varieties just waiting for the right antigen to present itself. Now, of these cells, only one is complementary to the particular antigen of the pathogen that has caused the infection. So only one of these T helper cells is going to be activated, and it's going to be the T helper cell that can recognize that antigen. So of all these, this one is going to be able to bind to this. So then what happens next? So that T helper cell with its CD4 receptor will bind that antigen that's been presented, and this will cause its activation. Okay, so the T cell, so let's just put that as a next stage. T cell with complementary CD4 receptor becomes activated. Okay, and this is part of what makes this response what we call specific, because it took specific interaction between the antigen and the CD4 receptor complementary binding to be able to select the right T helper cell that will be able to make the response. All the other cells, all the other T helper cells do not recognize that antigen and therefore would not be effective. Okay? Next, Once this T helper cell is activated, it then goes through many rounds of mitosis and some differentiation to produce 3 types of T cell. Okay, 3 types of T cell. One is more T helper cells of the right type. So again, a T helper with the same CD4 receptor. Another is what we talked about before, T memory cells. Again, it would have to bear the same receptor for the same antigen. Finally, I guess the most active of all the cells, T killer cells with the same receptor. Yeah? Now, what these T killer cells do is they're going to make their way, they move through the body, in the blood, lymphatic system, moving around the body, and whenever their receptor encounters, so this cell has been infected, right? So on its surface, it's presenting the antigen of the thing that's caused its infection. And so this T killer cell is going to come along, recognize that, look, this cell has been infected and I can recognize the antigen that's being presented, therefore I have just latched on to an infected cell. And when this interaction happens, the T killer cell releases chemicals or substances that cause the death of the cell it has attached to. Making sense? Okay. So, are we happy with the sequence of events that started from a pathogen being in the body to activation of the T helper cell, mitosis, differentiation, generation of T killer cells that will kill any cell infected with that pathogen? Okay? And that's essentially what this very large, so I'm just showing you one, but there's thousands of these cells moving through the body, identifying any cell infected with this pathogen and killing it. And remember that's what the job of the T cell branch was, to kill infected cells, so that these infected cells could not be factories for more pathogen.
What does cytotoxic mean?
Yeah, cytotoxic is just another way of saying killer. So yeah, it's cytotoxic, it's killing cells. Toxic to cells, cytotoxic. Yeah. Okay. So now, where do the B cells come into this? Now remember, we've still got free pathogens, so this pathogen could easily infect this cell. The T killer cell cannot do anything about this. So we need the B cells to do something about this. And so we have our T helper cells, and what the T helper cells do is, just like we had to select the right T helper cell, we now have to select the right B cell that can do something. So all these B cells, what they do is they are also antigen presenting cells. All these B cells, they act as antigen presenting cells. But all these different B cells will be presenting different types of antigen. Now, as part of this particular response, which B cell do we need to activate? This one, right? Because this is the B cell that's going to be able to act against the same antigen, and therefore act in a coordinated way with the T helper cells. Now, so what happens is that this T helper cell moves over here, or moves throughout the body, and what it does is, like in a very similar process, because it binds the same antigen that the B cell is binding, they undergo like some kind of a handshake where they recognize each other. And then the T helper cell releases a substance that causes the B cell to be activated, called cytokines. Okay, so there's two things here. The T helper cell has to undergo complementary binding to the antigen being presented. That's how it chooses the right B cell. But once it's chosen the right B cell, it then releases the cytokines that causes the B cell to become activated. Okay, so do mitosis, differentiation, then T helper cell via its CD4 receptor recognizes or selects B cell through antigen binding. Is that making sense? Okay, the T helper cell via its CD4 receptor recognizes and selects the B cell through antigen binding, ensuring that they are both acting against the same antigen. Okay, next is it releases cytokines to activate the B cell. And when this activation happens, I'm going to just move this, just going to get rid of it. Are we happy with this part? When this activation happens, the B cell then undergoes its own mitosis and differentiation to produce a number of different cell types. Yeah. First, it produces a cell type called a plasma cell. Plasma cell. And what plasma cells do is produce antibodies which are capable of binding to this particular antigen. Okay? So that's the job of the plasma cell. But it also produces, via the differentiation, it also produces B memory. Right? And they're not going to have an active role on this occasion, but they're going to hang around for future. Yeah? So in case this pathogen ever comes in again, these cells are going to be there to make a faster, bigger response the next time around. Yeah, because to undergo all this process is quite relatively slow. Yeah, and the first time around we can't make that big of a response. So we make some antibodies, but not a lot. So think of it as just enough to deal with the infection. But the first time round we will feel symptoms because this response is just about managing to keep on top of the infection. But via this primary response, we've generated this and we've generated that. So the next time round, quicker, bigger response. Secondary response. Okay, so B memory cells will hang around, T memory cells will hang around. They are kind of background for now, but they'll be important in the secondary infection. But for now, we've got T killer cells killing infected cells. We've got plasma cells that are producing antibody that will bind to the antigen on the surface of any free pathogen and thereby cause destruction via phagocytosis. Okay? I'll just clarify that last point in case it's a bit like, what the hell was going on there? So we've got a pathogen, and because it's got antibodies stuck to it, because these antibodies are kind of complementary, right? Now these antibodies from one side they bind the antigen, but on the other side they actually bind to a macrophage. So the antibody is actually helping the macrophage to extend its membrane around the pathogen. Yeah? So if the pathogen is covered with antibody, it helps the macrophage to engulf it and remove it.
So in the phagocytosis process, the membrane of the
Phagocyte
has a binding sites.
Yeah, good. For the other end of the antibody.
The things taken out, is it the antibodies or receptor sites?
What?
Yeah, those.
This?
Yeah.
That is antibody. Actually, yeah, let me just draw that in a more accurate way. So what's happening is that the membrane has binding sites for the antibody on this side. Yeah? It's got binding site there for the antibody. Binding site. And by binding there, it's helping it to extend its membrane around. Yeah? So the last thing that remains is, we just put here, only needed for secondary response there for the T memory, and again the B memory, secondary response. That's when they will have their effect. But you have to go through the primary response to generate the T memory and B memory cells. Okay, that's immune system. Yeah? Don't worry about memorizing it, worry about just understanding all the steps are, first instance.